Hi, I'm Daniel Bird. I'm a postdoctoral researcher working with Professor Vincent Harley at the Hudson Institute in Melbourne, Australia. I'd like to share with you some of the highlights from our recent endocrinology article, which explores the role of FGF signaling in gonadal development. In the Harley lab, we're interested in understanding the underlying genetic mechanisms regulating sex determination. That is, what genes are responsible for the choice of developing ovaries or testes. In mice, testicular development is dependent upon high levels of SOX9, which is initially triggered by upregulation of SRY. High SOX9 levels also require FGF9 signaling, which acts via its receptor Fgfr2 to suppress the ovarian Wnt4 pathway, a known inhibitor of SOX9. However, we wanted to know whether FGF9 was needed to also suppress a parallel ovarian pathway driven by FOXL2. We also wanted to specify which particular splice variant of FGFR2, FGFR2b or FGFR2c, was responsible for mediating the FGF9 signaling. Previously published work indicated that male FGFR2b knockout mice had normal testes development, yet male FGFR2c knockout mice are also reported to be fertile, suggesting that testes development was again normal. However, we found that 2C knockout mice on a CD1 genetic background showed disrupted testicular development and evidence of XY sex reversal, with most gonads resembling ovotestis, structures which include both testicular and ovarian material. Now, it is known in this field that when ovotestis develop, they can resolve into small testes and may still give functional sperm. This explains why the testicular phenotype in 2C knockout mice may previously have been overlooked. However, when we backcrossed onto the C57 black 6 background, which sensitizes for XY sex reversal, almost all XY 2C knockout gonads were sex-reversed ovaries. This is shown by absence of the male Sertoli cell markers SOX9 and AMH, and ectopic upregulation of the female granulosa cell marker FOXL2. This confirms FGFR2c as the isoform responsible for mediating FGF9 in the testis. We next asked whether testis determination requires FGF9 to suppress the FOXL2 pathway in the same way as it's needed to suppress Wnt4. To do this, we generated double FGFR2c FOXL2 knockout mice and looked to see whether testis development had been rescued. Interestingly, we found that embryonic gonads from XY double knockout mice were found either as rescued testis, ovotestis, or ovaries, with male and female cell lineage markers present or absent accordingly. This suggested to us a model whereby FGF9 signaling is initially required to suppress Wnt4, which establishes the ovarian cell fate, and then later is required to suppress FOXL2, which maintains the ovarian cell fate. Our mix of phenotypes in double knockout mice may therefore reflect variable Wnt4 levels and ovarian cell fate establishment between gonads. If you're interested to find out more, please read our article in full in November's issue of Endocrinology, and thanks for watching.